Disclaimer, disclaimer, disclaimer. The following hour of programming was conceived in the Big Bang. It has been under construction since before the pyramids were built. Contains information that predates the sun, and yet will consist solely of things revealed in the past week. If this makes your head spin, don't panic. This is a perfectly normal reaction for a creature that lives on a planet that is spinning, in a solar system that is spinning, in a galaxy spinning in our atoms too. Atoms spinning since before the first day began, spinning now and into the future and beyond that too. And all of it is spinning around a single solitary hour of programming, an attempt to give the cosmic flow of time and space some form of manageable conceptual context. This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kirsten and Blair.
Good science to you, Justin. Welcome everyone out there to another episode of This Week in Science. We are here with all sorts of sciencey goodness that we have culled from the universe of the internet to be able to discuss today on the show. And, I'm glad that Justin's not in jail, having been picked up in a Petaluma raid early this morning.
Narrowly escaped.
Narrowly escaped, exactly. But I hope everyone out there is ready for a big show full of amazing science. I brought stories. I'm going to cough here for a second. Excuse me. I brought some stories about CO2 that the government is pumping under the oceans, and some other stories about how crowds don't really follow the crowds, and some interesting cell suicide stories.
Oh, I have a stem cell suicide story. I hope it's not the same one. I've also got something that nobody probably knows about Clear Lake in Northern California. You know about the birds and the bees. Forget that. It's all about the fruit flies and the mice, as well as some hormonal eating. Hedonic hunger.
Blair, what did you bring?
Well, a couple of weeks ago, we talked about pigeons and how they had thought for decades that pigeons had metal in their beak that helped them sense magnetic fields. And then that was debunked recently. So now I have the latest theory. Should be interesting.
Awesome. Yeah. I'm actually really looking forward to talking about this story because this is right up my alley.
Yeah.
Birds, brains, magnets, all these things I love. I'm going to break things here. I'm moving my camera around like crazy trying to Blair in the shot because I still haven't figured out how to do multi-camera porting to Skype.
I forgot too. I've got a time machine story.
Ooh.
Yeah.
Good. I love time machines. We always love time machines. All right, so let's see. Let's get this show started, right? Let's get into these stories so we can get to all of them in the time that we have left. So, what is the government up to? Well, the Department of Energy has announced a study that it has been involved in. It's been working in conjunction with ConocoPhillips and a Japanese team to take clathrates and release methane from clathrates. So clathrates are these solid structures that form around pockets of methane. And the very nice way that they're described in this Ars Technica article by John Timmer is that they form at high pressure in water, which forms a cage-like structure around the pockets of methane. So methane hydrates, methane and water, otherwise known as clathrates, are found all over the place. There are these big methane reserves all over. And hey, if we can get the methane out of the clathrates, then it might be a great source of non-fossil fuel energy, right? Additionally, where these deposits are located, if you can get the methane out, maybe, thought a bunch of pretty smart people, maybe those pockets will remain and we can pump carbon dioxide in, and the pressure from the ocean and the sediments above where these clathrates were held, maybe that will keep the carbon dioxide sequestered beneath the surface of the earth. So they tried it, and they released the results of a project which took place off the coast of Alaska, and they found that they were able to get the clathrates out, and as they allowed the methane to escape, they pumped liquid carbon dioxide down to the space where the clathrates were held. And what they estimate is that those pressures should keep the carbon dioxide liquid, so it won't go into a gaseous form and escape. It should remain liquid, and it should remain trapped. However, now they've gotten to the point where the carbon dioxide is sequestered, the methane has come out. Now we need to know whether or not the methane that is released, whether they can control it well enough to gather the methane and not just release it into the atmosphere. How much of the process releases methane into the atmosphere? How much methane is released? And then how long will the carbon dioxide actually stay trapped? Is it going to be sequestered permanently, or is this just a very temporary solution? And is that carbon dioxide going to seep out slowly over time? There are a lot of questions that need to be answered. We don't know how permanent or even easy this solution is going to be. It definitely doesn't seem like it's going to be an easy solution. And the Department of Energy has put $6.5 million to further tests, and is requesting an additional $5 million from the government for budgets for next year. Kind of interesting. Yeah, I think it's a really fascinating. It's fascinating in the science of what they're actually trying to do in kind of replacing methane, which is not as long lived in the atmosphere as a greenhouse gas as carbon dioxide is. So we have methane, which is very active and actually has a very large effect within the atmosphere, but only for about 8 to 10 years, whereas carbon dioxide has an effect that lasts 50 years. Less effect, longer time than methane. So the question is, can we take this long forcing greenhouse gas of carbon dioxide? Can we sequester it beneath the surface of the earth? Will the pressures of the ocean and the sediments actually be able to keep it liquid, be able to keep it in place? Or are we going to see the sequestering just not really work? I mean, they think that the pressures will keep everything in place, but they don't know that they will. And so we need proof of concept experiments like this if we're going to actually move forward with these ideas of carbon dioxide sequestration, that everybody is like, woohoo, we're going to sequester carbon. You just pump it back under the surface of the earth and it's perfect. The earth can hold everything in its big pockets.
Yeah, and at what scale is that even a feasible solution? I mean, if we're talking about something that can sequester better than the ocean, right? The oceans of the earth, that 70% that covers the surface of the earth, it's going to have to be a really rapid and large scale sort of sequestration. I don't know quite how we're going to pull that one off, but it'd be nice to know we could if we had unlimited resources.
Yeah. Yeah, but it's the additional side to it, like I mentioned, is the methane that could be released from the clathrates. We know that there are huge deposits of clathrates around the world, and if we can get the methane out, then that would be fantastic for our natural gas reserves, for our hydrocarbon reserves that we'd want to use for energy. Methane burns really nicely, produces a lot of energy. Kind of nice. It's kind of nice.
It's also got, like, I think almost a magnitude of impact on climate for less duration, but has a larger impact in the atmosphere. So it's, I don't know.
Burying your problems in general, I think, is not a great tactic just in life.
Just burying
Bury
your problems.
There's always residual effects. It seems scary to me to just shove all the CO2 under there and hope that it doesn't affect anything.
If I hide my garbage under my other garbage, and then put more garbage on top of that garbage, it'll look like one pile of garbage.
Yep.
Yeah.
Yep, fair point. All right, what'd you bring, Justin?
I brought insect glands.
In your pockets?
I have insect glands, which are insect glands responsible for producing a host of secretions that allow the bee to sting, the ant to lay down trails, and from food to nest, are the focus of this study by Carnegie scientists. Specifically, the secretions from fly glands in the reproductive tract that help sperm survive and guide the sperm on the trip to fertilize an egg. So the gene that controls the development of these glands in fruit flies provides important information about gland development in all insects, as well as some potential clues for humans. Wow. So this is published in the Current Biology magazine. When a female fruit fly, receives sperm from a male fruit fly, lubricating secretions in her reproductive tract activate the sperm, store it, and guide it to fertilization. Without the aid of these secretions, the sperm would not ask directions and therefore never make it to the eggs. Alan Spalding and Jinjun Sun demonstrated that the gene in charge of regulating the development of fruit fly secretion glands is called HR39. It encodes a steroid receptor protein. Mutant fruit flies that lack this gene have no such glands formed in their reproductive tract and are therefore infertile. However, their formation could be partially restored with the expression of a mouse gene that encodes an analogous steroid receptor in mammals called LARH1. Mutant mice that lack this gene are also likewise infertile. So the work wildly demonstrates that even though it has been many, many, many millions of years since there was a link between mouse and fly, let alone any insect and a mammal, these similar genes are still in charge of at least some of the same functions, showing we're perhaps not that far removed after all. Secretions from reproductive glands in mammals are thought to also assist sperm in undergoing similar changes to facilitate fertilization. So they're looking at studying this process in general has been difficult, knowing that the functions in this fruit fly and the mammal are similar in this regard is going to allow them to do some further research. And, of course, in this, they're hoping the sort of end goal would be able to help fertilization in humans. But isn't that wild? You know, you take a gene from something like a mouse and introduce it to a fly, and it repairs the missing gene from the fly, or sort of serves a similar function. That is wild.
Yeah, I think any kind of genetic transfer of function or repair ability, especially across species, is amazing.
Across species, I mean, this is insect to mammal too. I mean, that's across a lot of species to get there. They had to pass up a whole lot of other species that are sitting in between them. Wow.
Yeah. And it goes to the point that genes are these little Legos, and you can just, I don't know if that's a correct analogy or an appropriate analogy necessarily, but they're like these little machines that you can take from one and put in another, and they have functions that are transferable, functions that make sense no matter where, which area of life you put them in. And the idea of synthetic biology, where we could mix and match and create our own organisms that kind of do what we want, or even reverse engineer to ourselves, humans, or even other animals, to to get rid of certain traits or figure out how certain traits work or to add new traits. I mean, there are all sorts of new ideas through, on the genetic side of things that as we studies like this just I think highlight that even more.
Yeah, we're all kind of running around with similar hardware.
It's the genes, man. It's the genes. It's the, what is it? The selfish gene hypothesis that really life is all about. It's all about replicating, reproducing the genes. It's not about you. It's not about me.
Well, and that's I think we talked about this before, pretty recently. That's like that's a main function of your brain, is to get the genes going to another generation and to keep you alive.
Yeah.
And then we do all this other stuff on the side. We create a society and make movies and read books and go out to nice restaurants or whatever we do. That's all peripheral, whatever. Yeah, do whatever you want on your time, but you're here to eat and reproduce. And that's what the brain demands.
That's what the brain wills you to do. It wills it. And maybe this is one step closer to the Jeff Goldblum movie, The Fly, right?
Yeah, could be. That was a good movie.
Could be.
Fantastic movie.
Yeah, exactly. Everybody, if you just tuned in, you are listening to This Week in Science. It is time for Blair's Animal Corner.
Blair's Animal Corner with Blair.
Everybody.
Hi, Blair.
So let's talk about pigeons, shall we? Pigeons, just a couple weeks ago, we found out that what we had thought for years and years helped them to be such good homers that we thought they were before, where they knew where they were all the time. They had kind of a GPS built in. We thought it was these magnet fragments in their metal fragments in their beak that responded to the magnetic fields that helped them navigate. But we found out that wasn't true. They were actually white blood cells. So that kind of brought us back to the drawing board on that. And so they recently at Baylor College of Medicine, this guy, David Dickman, did a bunch of experiments with pigeons. And I really liked, he had a really funny quote about it. He said, I can't find it now. He said that we're puzzled about, he used the word puzzled about the pigeons. And so even with this new finding that I'm going to talk about in a second, they're not totally sure about it. They have just preliminary stuff telling them they're still very much puzzled about this whole pigeon problem. Yes, be puzzled because I am. There it is. Because he was puzzled, so we should all be puzzled as well. Okay. So what they did is they put these pigeons, they were looking at neurons in the brain. They put these pigeons in a dark room, and they actually manipulated the magnetic field in the room. Mm-hmm. So they watched how neurons fired in the brain, and they found 53 neurons that responded to the changes in the magnetic field in the room.
Wow.
And so basically they found that these neurons work to make a 3D compass. They can figure out which direction they're going in, and they can also figure out basically longitude and latitude of where they are on the planet, which is kind of crazy. But so not only they know where they're going, but they know where they are based on these neurons firing in their brain. So, I mean, that's pretty interesting. It's a lot more complex than just metal in their body responding to a magnetic field.
Yeah, there was this, at one point, the idea was that if the magnetic particles were in cells by the beak, then maybe it led to some kind of a glow, adding like a color or something to the visual field that became stronger when they were in a stronger area of magnetic field, or that. But they had no idea of knowing, and that got booted out, like you said. And this actually makes a lot more sense, that there would be a network of neurons inside the brain involved in actually figuring out where the animal is in space. And so you have the visual system that's obviously going to tie visual cues into these other pieces of information. You have the vestibular system, which is involved in balance. And for us standing on the ground, we have one type of balance sense. But, could you imagine that you were always jumping over a trampoline, or always flying, doing a backflip?
Wait a second.
Would be a little bit different, getting some a little bit different information.
Wait a second.
From there, there's the what?
Wouldn't it, no matter what the source ultimately is, whether it was metal in the beak, maybe it's metal in the toes. If it is an external something that's triggering this, wouldn't it still have a center in the brain where neurons would be dedicated to translating that information?
Yeah. And so the big question is still, where is the receptor? They don't know. They haven't found where this information is coming yet and coming from yet. And, it's not from magnetic particles in cells in the beak. We know that now. So now the question is, are there other cells, receptor cells, that we still have to find? Did they have any commentary about that in the places you were reading?
Yeah. So basically, they just said that the MR cells in the brain, they encode a geomagnetic vector, and that they computationally derive the bird's position and directional heading from these MR cells. But so it was just a preliminary experiment. So this was just basically step one. So they put these birds in the dark room and they figured out which neurons were working, basically.
Yeah.
So
and you have some neurons that are going to respond to polarity more strongly, and other neurons that are going to respond more strongly to different strength magnetic fields, and some that are going to respond more strongly to direction of the magnetic field. And then the network, and they only measured 53 neurons of the network. And, that's not the network. That's a very small part of the network that's in the brain. It's just they happen to be able to stick their needles into the brains of the birds and get them close enough to these neurons to be able to record from them. And so there's probably a very intricate, dedicated network that, as you said, computationally, through the strengths and the relative firing patterns of these different neurons, puts it all together so that the bird can think, the magnetic field, I need to head west. I'm going, or it's springtime and I need to go north.
Yeah. And I find it so interesting because, growing up in a city, you see pigeons around and you kind of just take them for granted and you think they're flying rats or whatever. And really, they're complicated little birds. We've been using them for hundreds of years for homing and sending out messages and all this kind of complex stuff. There's so much going on in that little bird brain that we don't even know about. It's fascinating to me.
Yeah. And so I always hope that the next time that somebody looks at a pigeon in a city and is like, rats with wings, blah, that they just actually think about it for a minute and go, oh, but rats are really smart.
Yeah.
That pigeon's really smart too. Yeah. And these, and something that I think is always very important to consider is that animals that are succeeding in an environment are, they're going to be very smart for that environment, very well adapted. They're going to have the ability to be able to have plasticity to the way that they think and that they act and behave in certain situations. And like you said, pigeons are a diverse group of birds that we actually have been using for a lot of functions. And especially people who might breed and work with homing pigeons, you've got to be aware of the complicated tasks that they're able to perform. You could take the bird, put it miles away, and they'll be able to get back to where their home roost is.
Yeah. And so, I know for a long time they thought that even just recently, I went somewhere where they had homing pigeons. When I was in Israel, they had homing pigeons there. And they were talking about the metal in the beak and how when they're born somewhere, they can always find their way right back to where they were hatched because of the metal in the beak. And they were going on and on about that. But so not only they know where they are with whatever's going on in their brain, but they know what home is, which is such an interesting concept because not all animal species can do that.
Yeah. Yeah, and that's a really interesting point. In some animal species are. So you have sea turtles. There's some species of butterfly that are able to do that.
Salmon, of course.
Salmon, yes. Salmon, not so much with the magnetic sense, but the olfaction. And birds as well. And there are not all species of birds even, but many species of birds are able to remember where home is. And the interesting thing is that there's a very critical period during their young years where they imprint on the sensory information that's available to them in their environment. And so if you have receptors, which we still haven't found in the birds, but if you have this network in the brain for translating the magnetic information and actually turning that into home feels like this.
Right.
This is what the magnetic field of home feels like.
Yeah.
At the same time, they're imprinting on that's what daddy looks like. That's what mommy sounds like. That's what, they're listening to other individuals in the environment. They're going, this is what food looks like. And they're imprinting on all that information.
Yeah.
So they can put it together. And then for migratory birds, they go on their first migration and they follow that magnetic sense and they just kind of follow their nose until they get to the place where they're supposed to be. And then they imprint again on that new location so that they can get back there again in the future.
Yeah. And you can then, of course, you start thinking about humans because it always comes back to humans, right? That, there's this whole home sweet home thing that we have built in as this kind of human quality of feeling at home somewhere and comforted. And actually, that's a pretty basal animalistic thing, is to have a home base that is a safe zone. It's actually interesting that so many animals have that as well.
And in birds, it comes back to magnets.
Yeah.
Somewhere. Yeah. But it is interesting. I was looking at one article, and the researcher said, the various areas of the brain that were involved. There were 4 areas: the hippocampus, the vestibular region of the brainstem, the hippocampus. Did I say the hippocampus already? I said the dorsal thalamus and the visual cortex. That's what it was. And he said, if I were to build a 3D compass in the brain, that's exactly the areas of the brain that I would use. Well, great.
Well, there you go.
It's a good way to think about it. I don't know. Let's see. Oh, I had one more story. Should we have one more story before we go to the break?
Yeah.
Yes. So crowds. There's been there for several years. People have had this idea that if you, and I think the original experiments were done like in the 1960s or something, that if somebody stops and stares at something, that people will stop and to see what that person is looking at. And in stopping to see what the person is looking at, they will then get more people to stop to look at what the original person stopped to look at. And so it's been thought for a really long time that there's some kind of a tipping point or a threshold, at which point basically it's like a flash mob for staring at whatever, it's just like everybody's gonna look. But nobody's been able to really replicate this idea experimentally. And so researchers just published some work where they were studying this phenomenon in Oxford, and they had a few different scenarios. One was in a shopping area, and they put a camera up, and they had assistants who would stop at a given point and just stare and stare at the camera. And then they would check, and and the assistants were set out in different sized groups to see if the size of the group that stopped and stared at the camera had any influence on the number of people who stopped and stared additionally. And so in one area, they were in a shopping area. Another area, they were, I think, in a train station. And so there's the one experiment where the people stopped to stare at a camera. And then at the train station, they did the stopping to stare at a camera. But then in another version of it, they had a couple of guys with that looked suspicious, stopping and looking at stuff as if they were trying to map it. And so basically making them appear as if they were going to do something that was wrong or bad. And so in almost every situation, they found that people did stop and stare. However, they couldn't get that threshold level. They absolutely could not get people to really stop en masse and stare at the camera with this group.
That's such a weird. That's sort of like a weird experiment to me.
Yeah.
For instance, it was like I sort of see it as a law of attraction. You see other people doing it. Maybe it's safe to stare at something, so then you look because other people already are. But you have to have something interesting to look at in the first place. Like I would redo the experiment with somebody who's just like a mediocre juggler, right? And just that's all they're doing, and have nobody stop and so just see how many people actually stop and do more than take a couple glances at this mediocre juggler. Yeah. Versus you throw a crowd of like 5 people like actively like, whoa, wow, and then suddenly I bet you build a crowd of like other people coming, oh, wow, oh, look, there's somebody juggling. These people are enjoying it. I'm going to enjoy some juggling too. Wow,
right.
But
that's not necessarily that's not necessarily a thing. It's more if they just wanted to know if they could get people to stop
at nothing,
to stare, to see what people were looking at. Right. What is that group of people looking at over there? And and they found that the slower people were moving, they were more likely to stop and look as well. The more quickly they're moving, the less likely they were to stop. And that kind of makes sense. If you're moving more slowly, you're probably, not dedicated to getting somewhere at a certain time. And so you're more looking around anyway. And, oh, what's going on over there? Okay, I'll check that out. They also found that people who were closer tended to stop and stare more than people who are further away, so that the further away you were from the starers, the less likely you were to start staring as well. The interesting result, though, that came out of this, though, like they were trying to replicate this threshold of people being able to stop and stare, and that just didn't happen. But the really interesting result that came out of this study is that they found the suspicious people in the train station got a lot of attention. And they got a lot of attention from people really close to them, the same as in the shopping district and with the people looking at a camera. But instead of staring where the suspicious people were looking, people close to them averted their eyes and tended to look away and pretend not to see them. Huh. So it's just a fascinating result. And this lack of attention, they report, it was driven almost entirely by males. And the authors think that maybe because the individuals were suspicious and male in that were the assistants in the train station, that they think that males would think of staring at those individuals or in the direction that those individuals are staring, that would be taken as aggression. And so maybe males close to those individuals would be less likely to aggressively do an aggressive behavior like staring, and more likely to just look away and keep going, because maybe those suspicious individuals are aggressive and maybe they would be apt to do something, or that something a confrontation would result. Yeah, But it's an interesting result that in the case of a suspicious or confrontational type of thing that people, especially men, tend not to look in that direction unless they're far away. And so it was people who were further away from the suspicious individuals who were more likely to actually stare.
But then there's more to this than that perhaps isn't exactly being clarified. Because they must have done something provocatively to make these people seem suspicious and or dangerous. Because I don't care where you put me on the planet. If I'm staring suspiciously at something like this, people aren't freaking out. You know what I mean? They're just not,
Yeah.
So I found this really funny, actually, because when I was in high school... I could even do this,
and people wouldn't...
Yeah.
I was kind of a strange child in high school, and me and my friends would go over to Union Square, and we would pick a spot out on the street and stare up at the top of a building across the street, and we'd see how many tourists would stop and look.
Oh, that's fun.
So
you did this.
We did it. And actually quite a few people did stop. And eventually we'd amp it up and we'd start pointing and scratching our head. And we'd be looking at nothing. And we just thought it was funny to mess with the people walking down by Union Square. And lots of people did stop and look.
Did you ever get like a flash mob of people all looking, or was it mostly like stopping and looking and passing by?
Yeah, it was mostly just stop, look, pass. But most people would look. If they were maybe in a suit going to a job, resident, they're not going to look.
Yeah.
But the people that were just kind of walking around, checking out the sites would definitely stop and look.
I think that's fascinating. That's really interesting. So staring, providing us with lots of information about how humans think and how we act in groups.
I still think they needed a mediocre juggler to really figure out what was going on there.
Absolutely. Well, I think that we need some above average outro music and a station break. So we will be back in just a few moments with more This Week in Science. Stay with us. Audible.com is the leading provider of audiobooks with over 100,000, used to be 75,000, but people keep writing books and people keep recording books and their library just keeps getting bigger. So 100,000 different titles in a variety of different genres, including science, which Twis absolutely adores. And we've found many books over the years in the science topics, nonfiction science topics in the Audible library. And we know that you'll find books that you'll love too. You can start a free trial today. You can get any audiobook download of your choice for free. All you have to do is sign up at audiblepodcast.com/twist. That's audiblepodcast.com/twist. If you go there, use that URL, audiblepodcast.com/twist right now. You can just get started listening to audiobooks and enjoy now. Twis also has merchandise you might enjoy, so head on over to twis.org. You can purchase the 2010, that's right, I said 2010, we're a couple years behind, Science Music Compilation CD. And if you need clothing, World Robot Domination t-shirt. That's right. We have t-shirts and CDs that you can buy. You can buy, you can listen, you can wear, and enjoy. It's twist.org to head over to purchase that merchandise. And Twis is also supported not just by the merchandise, but by donations from the likes of you, our listeners. Your donations pay for hosting, bandwidth, contractors we need to hire, and very occasionally fun things that we try to do for the show. The very first compilation CD was produced as a result of donations. We appreciate any amount. $2, $5, $5,000, $1 million, whatever you are able to give, we do appreciate. You make this show possible. We currently accept donations using PayPal on our website, and we've made the process really easy for you by putting donation buttons on each show page on the website. So once again, go to twis.org. You can listen to the most recent episode, comment on the show as you're there, and while you're there on the page, there are little pink donation buttons. Click one of those and make a donation. We thank you for your support. We really couldn't do it without you. There are things falling and breaking. What's going on?
Everything is under control.
Yeah.
Everything is going to be okay.
That's right. Welcome back, everybody. This is This Week in Science. What did you want to talk about
Somebody
now,
in the chat room, real quick. Somebody in the chat room said, you learn better by reading than you do by hearing things. Maybe true for you. Everybody does have a different way of learning things, though. So not necessarily true for everyone. It's not. It's hard to generalize the way that people learn. People learn in lots of interesting ways. Some people, it helps them to be actually physically active, bouncing around, doing something while reading and hearing at the same time. Where some people can just read stuff, retain it forever. For some people like me, I read stuff and forget it immediately. But if I hear it, it's locked. It's good. I'll keep it.
Yeah. What story did you want to talk about? What's going on? What science did you bring?
Did we bring the same? Did you bring a story about stem cell suicide?
Don't do it.
Did you? No. You want me to? I could.
Do you remember that song? Teen suicide. Don't do it. Stem cell suicide. Don't do it. Well, anyway, we do need stem cell suicide. Probably very similar, but go ahead.
Okay, those revered noble cells that give rise to every cell type in the body just got another badge of honor as scientists witnessed them committing the ultimate act of altruistic self-sacrifice. If they suffer damage of any sort that makes them a threat to the developing embryo, they commit suicide. They kill themselves. They end their life, their existence, which is very unusual for most cells in the human body. Thankfully, they don't all do that, at least not as quickly. So this finding offers a new glimpse into the private lives of stem cells that could help scientists use them to grow new neurons or other cells to replace those that have been lost in patients with Parkinson's and other diseases. Despite the huge potential of stem cells for therapeutic use, very few people have actually investigated their basic biology, says the study's senior researcher Mohanish Deshmukh, PhD professor of cell and developmental biology, University of North Carolina, Chapel Hill. These results could have significant implications from a therapeutic perspective. Of all the important things our bodies do, staying alive is clearly at the top of the list for most of the cells. But a cell's ability to die when something in its DNA goes wrong is also critical. Cancer is basically runaway cells where there's been damage to the DNA of the cell, but instead of killing themselves off, they replicate. Unchecked even, and become turned into tumors and the like. Deshmukh and his colleagues discovered that stem cells are extremely sensitive to DNA damage. Things that can be caused, that causes damage, chemicals, radiation, viruses can cause DNA damage. And the experiment showed that virtually 100%, wow, that's amazing, 100% of human embryonic stem cells treated with a DNA-damaging drug killed themselves within 5 hours. As compared to
5 hours.
5 hours, which in the comparison is other cells in the body might be programmed to do so also, but it would take in the neighborhood of 24 hours or more.
Longer.
Or longer, correct. So, yeah, an incredibly rapid rate of death, says Deshmukh, who also is a member of the UNC Neuroscience Center and Limburger Comprehensive Cancer Center. So he's looking at this from a few angles. The hair trigger suicidal response is an important adaptation for embryonic stem cells, said the researcher, because the slower response would allow DNA damage to proliferate and harm the embryo. Mutations that develop in these cells could be catastrophic for the developing organism, so it would make sense for these cells to be rapidly eliminated. So the key here to the quick response by the stem cells is a protein called BAX that the researchers found. In most cells, BAX is kept in a very inactive form, waiting for a long chain of events to occur before the BAX is brought into play. And the embryonic stem cells, team found BAX standing at attention in its active form in the Golgi apparatus. Golgi? In the Golgi apparatus, a part of the cell that processes and modifies proteins. Says Deshmukh, what these cells do is very clever. Too bad intelligent design was taken for something else, other meaning. We could have clever design. I guess that could be taken back, right? They have activated BAX, but they've also parked it in a safe little compartment. If the cell detects DNA damage, BAX zips over to the mitochondrion, the power plant of the cell, where it signals other proteins to shut down the cell. So they have these built-in safeguards against hair trigger activation of death, but the pre-activated BAX housed in the Golgi
apparatus.
In the Golgi apparatus, keeping the protein from accidentally triggering cell death when not unwarranted.
Yeah, I think this is really interesting that most cells, like you mentioned, the BAX is lounging somewhere. It's not ready to go. You know, it's like a centurion or some kind of warrior, a knight that doesn't have its equipment, its armor put on yet. You know, its sword's over in the corner, the shield is somewhere else, whatever. Okay, it's going to take me so long to get ready, right? But the BAX in these stem cells is ready. They've got all of their stuff on. They're ready to go. They're amped up, but they're held in place. And so if the signal comes that that there's damage, that the cell needs to commit suicide for the health of the organism, then bam, they're released. They get out there, they can act, and in 5 hours cause the death of the cell versus day or longer to get that process going. I just think it's a really, really interesting difference. And the fact that they also found in this that after the cells, these stem cells start differentiating. So once they start deciding that they're going to be a certain type of tissue, so you have the progenitor stem cells, which these are, and they say they're like 50 of these cells, a very small number of them, but they become any cell type in the body. They turn into everything, and their future is not yet decided. But the minute or second that they start differentiating into, this one becomes an epidermal stem cell, so it's going to go on to become skin, or maybe a neuronal stem cell. It'll go on to become brain cells, or a muscular whatever it ends up deciding to be. At that point, the BAX is then put back to rest, and it from that point forward will not affect the suicide of the cell as rapidly as it was when they were just these little embryonic stem cells.
Yeah, totally. And dude man in the chat room says we shouldn't anthropomorphize evolutionary processes. I would actually reverse that and say we totally can do that. In fact, we might want to evolutionarily process what humans do and our behavior, because it's actually we're very similar to things like evolutionary processes and the behavior of bacteria. We're not that different. So I don't think it's a problem of anthropomorphizing a process but more just sort of showing how little has changed from the very building blocks of life to what we do as humans. It's really not that different.
Yeah, the interesting thing about this and the implications for what it'll allow researchers to understand is knowing that there's a difference in the way that this one protein, this one protein, this executioner protein, is made ready in the embryonic cells during a very short period of time during organismal development versus the entire rest of the development of the organism. That'll make I think it'll allow researchers to have some real insights into when and how certain tissues will get shut down, what certain types of damage lead to apoptosis and cell death, and why that's important. There's a lot more that needs to be learned. I think it's fascinating.
Mm-hmm.
Not anthropomorphizing about it at all, but it's just a really cool understanding how we are made is just fascinating.
Then somebody in the chat room also threw out the word consciousness. It's just the stuff that happens in between evolutionary processes. It's
right.
It's over here on the side. It's not that significant to evolution in humanity, really. It's just,
yeah,
it's
and
this
when we talk about apoptosis or programmed cell death or cell suicide, there are multiple reasons why apoptosis takes place. And at the very beginning, if you have DNA damage to the organism that's in those very progenitor cells that go on to become every cell of the body, that DNA damage would get propagated throughout every cell of your body, and that could cause some real problems down the line if that organism were allowed to make it all the way to the end of gestation by the biology. But these natural controls are in place, and the cells themselves go, we're broken. This is not going to be good, and just stop it at that point and kill themselves off. In other situations, you actually need pruning to take place. Just about every tissue in the body, you need a certain amount of programmed cell death so that the tissue will develop properly and will remain healthy. Otherwise, you end up with cancer.
Mm-hmm. And to take it to the human level, it would mean that nobody had ever died ever in the history of the world, and housing prices would be insane.
Yeah, really insane. And along these lines, there was another study that I thought was really interesting, not stem cells or backs, but published in Science, a study allowing us to understand telomeres. So telomeres are the ends of our chromosomes, and telomeres are thought to be or have been implicated in the aging process. So as long as your telomeres are maintained, then your cells are fine and they don't end up undergoing apoptosis, or usually. But if your telomeres end up being shortened, and there are certain metabolic processes that can shorten your telomeres, free radicals can damage your telomeres, along with a number of other things. And just as you get older, your telomeres, the telomerase that keeps your telomeres nice and long and healthy, it doesn't work as well, and so the telomeres start to shorten. So there's this other aspect of telomeres that I hadn't heard of before, but there is a cap over telomeres. It's called shelterin. So shelterin are this group of proteins that form a cap over the telomere, and by forming the cap over the telomere, they actually aid it in being protected from some of these DNA repair systems that go wrong at times and shorten the telomeres and end up leading to more rapid aging. So these proteins, 6 proteins called shelterin, researchers wanted to know how they work and what exactly they're protecting the telomeres from. And so they put together a group of experiments where they created mice that were genetically modified to remove each of the shelter-in proteins. So the 6 proteins, in different strains of the mice would remove the different proteins. And so they also had, they managed to remove the entire group by getting rid of the 2 members that anchor the rest of the telomere. They had to knock out other proteins that would fuse the uncapped telomeres together to allow them to figure out what happens to the naked ends. And so the quote from the researcher said, in respect to breeding the right mice to be able to do these experiments, it's an awful experiment to do. I wouldn't have subjected anyone else in my lab to it. So I guess it took a lot of work and a lot of time to actually get this research done. So what they ended up finding is that the uncapped ends of your telomeres, so if shelter-in is removed, the uncapped ends are vulnerable to 6 different repair mechanisms. And one of the overlooked pathways is an alternative way of joining 2 ends. One involves cutting back one of the exposed DNA strands. The researcher says, our work describes the full spectrum of events that could happen at chromosome ends that have lost telomere protection. There aren't 4 pathways, but 6 that pose a threat to the integrity of the chromosome end and that telomeres must protect against. And so once the telomeres are gone, then the naked DNA is exposed to these 6 repair mechanisms that can slice and dice and stick things together in ways that the chromosomes are not supposed to be put together for them to work properly, and for proteins to be produced, for genes to be translated properly. So cellular aging, as we get older, the aging process, or even accelerated aging, there is a disease in which people become old too rapidly, and a lot of this has to do with the telomeres. And so, if we can figure out how to protect the telomeres longer, then we find ways to maybe expand lifespan, to slow down aging in people who have this disorder, or to, just contribute to better longevity. Yeah.
And greater housing costs.
Oh, come on. We're all going to live in high rises in cities eventually.
High rises in the
And
clouds.
our food will come in pill form. We'll be fine.
The replicator, right? Yeah.
Oh, my goodness. Well, we're talking about living longer. Maybe we'll live long enough to hear the end of this story. This is a time travel story that's taking place at the bottom of Clear Lake in Northern California. Now, if anybody's familiar with Clear Lake, unlike the name, the lake is not really so clear. I don't know that it's really much of a destination spot, really. I don't know if anything goes on in Clear Lake. I know they had, aggressive pike.
Canocti Harbor. They have music shows and stuff there.
Okay.
And I've been there. I've camped there, actually. It's
Clear
nice.
Lake? Clear Lake. Oh, it's a wonderful
nice.
place.
It used to be clear, but it's not so much anymore. Yeah.
Listen to this. This is something I'm sure you did not know about Clear Lake.
I'm
I
sure.
certainly didn't.
Go.
The lake sediments at the bottom of the lake are among the world's oldest. Not California's oldest, not North America, the world's oldest sediments. They say these contain records of biological changes stretching back as far as 500,000 years. Clear Lake. Who knew, right?
Yeah, that's awesome.
So they're doing this drilling, part of a multifaceted effort at UC Berkeley to determine how Earth's flora, fauna respond to past changes in climate in order to improve the models that they have now for projecting how life in, Northern California, but then Earth in general, will adapt to the changes due to global warming doing all this, of course, in Clear Lake, of all insane places for this to be happening. Yeah, it's unusual in having survived. It survived the advance and retreat of glaciers that scoured and obliterated most lakes outside of the tropics, including the large lakes of the California Sierras, like we think Lake Tahoe or the Great Lakes, or, whatever lake you can think of, probably hasn't actually existed that long. Somehow, Clear Lake, not a destination over the last 500,000 years, even for glaciers. So there was a previous coring done there where they drilled down by the U.S. Geological Survey back in 1973 and again in 1980. And it revealed lake sediments 500,000 years old. And there were a few breaks in the continuity as they were going down. But they found an upper arm of the lake where one of the drillings took place, where it was a continuous core, at least going back 130,000 years. Why is this important? Well, there was a glacial change 130,000 years ago. So the original survey, they sampled cores once every meter, once every 3 feet or so. The researchers here are going to try to do it every centimeter. That sounds like, right? That sounds like it's going to be pretty intense. But a centimeter layer is going to equal about 10 years of time. So this should much better resolution than the first coring samples and give them a pretty clear look at the Clear Lake history. So they're going to, they've got a whole bevy of scientists. They're going to use isotope and chemical analysis as well as carbon dating. There's going to be, they're going to have pollen grains are going to be checked, charcoal, freshwater organisms in different times. And so what they're kind of going to do, they're also going to be matching the charcoal stuff is actually more for pretty recent times. It's going to be Native Americans because they did some sort of burning to help acorn trees produce more acorns. I'm not sure how that works. They also have lots of mammals and fossilized life forms from caves in the surrounding areas. So they're going to sort of match that up to what was going on backwards in time.
Yeah.
Really interesting. And so they're looking also, there's a period to Earth of, 113,000 years ago when it was warmer than today.
Mm-hmm.
And so they want to see what was living then, how it changed as the temperature changed. And that might give us a pretty good idea of what California, at least, is in for as we get into a region of somewhere between 2 to 4 degrees difference heading into the future.
Yeah, I think I love the idea of being able to go back to the past, kind of tell the story of what was happening at a particular time, who was there, what kind of plants, animals, what the environment was like. And so we know it was warmer. What was the vegetation like? How, maybe what were the animals like? What kind of stuff can we pull out of the information about it? And like you mentioned, there are caves and other things. So what kind of paleontological or anthropological information can we pull out of everything that could have happened around a particular time and then tell that story about, okay, what's going to happen in the future? How did people live in the past? How will they live in the future?
Yeah, they got 16 faculty members working on it. Paleontologists, pollen experts, botanists, ecologists, climate modeling experts. It's going to be a pretty all hands on deck kind of a study. They're also going to be able to, I guess, measure the amount of oxygen in the atmosphere over these times. It's going to be a really interesting study when this comes out.
Yeah. Yeah.
This isn't even
Clear Lake.
Yeah, I know, right here, right? One of the oldest reserves of lake sediment in the world.
Nice.
Yeah.
500,000 years. And we're not even getting, as far as these cores are going, they can go back 500,000, but they're just going back 130 or so.
Yeah, well, because that's where they're going back, I at that degree at least. That's that decade at a time.
A centimeter, yeah.
Right. I suppose they could collect more, but they're really curious about this period because they know it was warmer today. They're also going to look at a period about 13 to 11,000 years ago where there was another rapid change in temperature, almost at what we are currently experiencing, or we've currently experienced over the last 50 years. So they're gonna look at all those sorts of data points and see how dramatically or not dramatically the life forms changed in that area.
Nice. I'm excited. I can't wait to get, it's going to be a while, but I can't wait to get the updates. I'm sure the data and the analysis will pour in. Final story, facial recognition. Love facial recognition. Isn't it just this wonderful technology? Facebook is like, hello, is this you? In a photo, and you're like, no, leave me alone. Google's trying to do the same thing. Facial recognition for photos is very common in social media, across the web. Additionally, law enforcement is using facial recognition to be able to identify. Facial recognition probably got the biggest start in Las Vegas, trying to identify gamblers who have problems in the casinos and have been kicked out of various casinos. But now a new use for facial recognition analysis is in art history research. And the University of California, Riverside, some researchers have gotten a startup grant from the National Endowment for the Humanities, and they're going to be trying to use facial recognition to be able to identify individuals across different works of art, to be able to see if technology, and I quote, can add a new body of information that has an objectivity to it, as opposed to the bias that humans would potentially give. And so they say that their first round of testing is going to begin on the known death mask of Battista Sforza. And she was a 15th century Italian duchess. And they want to compare it to a sculpture that's attributed to her, but they don't actually know whether or not the sculpture is of her. So this will be the first test. So we know what she looks like in this painting, not painting, but in this death mask. And so can we take what this death mask looks like and map it to the sculpture and be able to see if the technology can identify the person and say, yes, it is, or no, it's not. And so this may help, if this works out, this could be the kind of thing that could help with mysteries of, there are lots of historical paintings of mysterious people, and who are the people in the paintings? And maybe this kind of thing could help identify individuals throughout history who have turned up unexpected in paintings or in sculpture of various sorts. And so they're going to be using the elastic bunch graph approach. What it's called is first described by a scientist in 1999, can solve the problem of image variation with respect to facial expression, head position, pose, and size. So say the head is turned, say it's up, say different expressions. Not that people listening to the show can actually tell that I'm making different expressions, but your brain is able to identify my face, whether or not I'm slightly turned away from the camera or I'm looking straight at you, I'm smiling or I'm frowning or I'm scowling. Your brain can determine that the person in front of you has not changed. This is the same person, just with a variety of poses and expressions. And so that is the problem of facial recognition, the crux of it, being able to identify an individual across all these different potential variations that could come out of a face.
I think it would be kind of fun if they, instead of worrying about how specific it was, left the identification ability of it kind of vague, kind of loose, and you could then track the ideal of artists over time, the image of beauty, what a beautiful woman looks like. What does a beautiful woman's face look like? And you could track it throughout art history as it sort of morphs and changes over time. That to me would be really interesting. You know, if
So
you
a
sort of
little
had
bit more, a little bit vaguer as opposed
like
to
woman A, B, C, D, and E, and see where she pops up in art.
very interesting.
Mm-hmm.
Yeah. Do you have anything else? I think we're at the end of the show.
Oh, I'm good.
You're good.
I'm good. There was another story here, but it's more about how the brain gives you drugs if you eat. It's
Well, we all knew that.
Yeah.
and we appreciate it. I like eating, and I like the drugs that the brain gives me as a reward for eating. Makes me happy, and I like to sit on the couch afterwards. No, but that's it for our show today. Justin, you're so far away in Petaluma.
I know. I'm closer than I normally am, aren't I?
Maybe,
Doesn't that
yeah.
make me closer?
Yeah, I guess, yeah. It's true. But will you be back there next week?
I will be back here again next week, as always.
And so will I.
More sciencey news.
Yeah, good.
Hooray.
Excellent. Next week, we'll be back.
Yes. Oh, yes. Hope you enjoyed the show today. Twis is also available as a podcast. You can just Google us in the iTunes directory. If you have an Android phone, you can look for Twis for Droid in the Google Play Marketplace. That's Twis, the number 4 Droid. And we also have an iTunes app. Just look for Twis in the iPhone app directory.
Yeah, iPhone. That's right. But for Twis-
TWIS. We're there.
And for more information on anything that you've heard here today, our stories, show notes will be available on our website, twist.org. Thanks to the lovely and talented Blair Baz.
My pleasure.
We also want to hear from you. So email us at kirsten@thisweekinscience.com or justin@thisweekinscience.com.
Be sure to put Twis, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also get a hold of us, shoot us notes, follow things we might be talking about at Jacksonfly, at Dr. Kiki, at Blair's
Menagerie.
Menagerie.
Blair's Menagerie.
Yeah.
And Twist Science.
Yes.
And at Twist Science. We're all over
absolutely.
the Twittosphere at the moment. There's actually 4 of us there, even though there's only 3 of us here.
Yeah.
That's right. And we will be back here next week, and we hope that you will join us once again for more great science news.
And if you've learned anything from today's show, remember,
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 laundry list of items I want to address. From stopping global hunger to dredging Loch Ness, I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science, This Week in Science, This Week in Science, 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 science. This Week in Science, This Week in Science, This Week in Science, 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 science. This Week in Science, 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 This Week in Science.